Hello everybody, welcome to Unit 4 Biology Area Study 2. Today we are looking at genetic changes in a population over time. So we're going to be looking at changes in gene pools, selection pressures, genetic drift and gene flow and mutations. We're going to be looking at selective breeding programs, and we're going to be looking at bacterial resistance. So we'll make a start with genetic drift and gene flow. So gene flow is basically looking at migration, and it can occur very quickly, unlike selection that occurs usually over numerous generations. So when we talk about gene flow, we can talk about alleles coming into a population or alleles leaving a certain population. So coming in, we call immigration with an I, and going out, we call emigration with an E. In terms of genetic drift, there are two types of genetic drift that we do talk about, and that is bottleneck and founder effects. So it doesn't favor one allele over another like natural selection might, and both alleles are equally subjected to being affected in genetic drift. So whatever alleles might be involved, the alternate forms of gene are involved, they might be equally subjected to being affected. So if we think about a population as well, a smaller population will probably have a much greater impact or be greatly impacted by genetic drift, whereas a larger population, there might be more stability there because there's more variability and more variation, and so they might be less affected by changes that may occur. So looking into bottleneck effect, it basically occurs when the size of the population is going to be drastically reduced, okay? And it could be due mostly to maybe a natural disaster. So something like a bushfire, a flood, predation, a disease. It's where there might be like a few survivors that are left over. And those few survivors that are left, they're the ones that are giving rise to that next generation. So that new population might not actually be an accurate representation of that original population. The population will rebuild, okay, but it's going to have potentially a very different allele frequency compared to the original population, and therefore reduce genetic diversity as well. So we've got a diagram here that sort of shows our population numbers. Some bottleneck impact event has occurred. Okay, our population drastically reduces, and then over time that population builds up, but the variability in that might be a lot less than that original population there. Founder effect, however, is basically where a new colony is created by a small group or a small few members from that larger group. So that new colony that might be created, okay, they might very well have a reduced genetic variation, and it would be a non-random sample of the original. So say this is our original population, okay, a small group of those decide to leave, and that's called that founder group, and they will create their own population, and so the variation in that population as well will be substantially lower compared to the original. So they might evolve differently to what the original population was. And a lot of the time, founder effect might cause high incidences of inherited disease as well. Looking onto the changes that this can have on genetic diversity, so both bottleneck and founder effect, they do decrease genetic diversity within a population. So genetic drift reduces genetic diversity in a population through the random removal of alleles, and so the way through each mechanism of genetic drift reduces genetic diversity as well. So gene flow can introduce or remove alleles from a population, and that can increase or decrease the variation that may be there as well. So here with bottleneck effect, we can see that the consequence is reduced genetic diversity through the removal of alleles because of some random event, so a natural disaster, whereas founder effect is a reduction in that genetic diversity because of the new population with an unrepresented sample of the original. We can also see here, this summarizes the effect of genetic drift and gene flow on genetic diversity as well. So we can see with genetic drift, our bottleneck effect and our founder effect both decrease genetic diversity, whereas with gene flow, immigration and interbreeding will increase genetic diversity, but emigration, where our alleles are moving out, of the population that will result in a reduction of the genetic diversity. Another way that can create some variations is through mutations as well. So a gene mutation is basically any change in the genetic material making up a gene, and this might commonly form a new resultant gene. So this might alter the encoded instructions that are in DNA as well. And there's 3 categories of mutations that we look at. They are point, frameshift, and block mutations. In terms of point mutations, they affect one single base, okay, and they can do that via substitution. So an A might swap for a T, insertion where we have an additional base, or deletion where we have a removal of a base. And it can occur in structural or regulatory genes, and there's 3 impacts that we talk about having when there's a point mutation. So a point mutation could be silent, so it means that the amino acid that might be encoded for is the same. So even though there is a change in the nucleotide, an A or a T or a C or a G, whatever the mutation is, it might still result in the same amino acid. And so that's silent because it's not going to affect the protein. We have also what we call a nonsense mutation, and that basically, instead of the DNA coding for an amino acid, is going to code for a stop codon. So that will stop that particular protein from being made. And a missense mutation is basically where the amino acid that was meant to be formed has now formed a different amino acid because of the code. And so this could be conservative or non-conservative. So a conservative missense mutation is where the change in amino acid is resulting in a similar amino acid, so the change can be tolerated. But a non-conservative missense mutation is where the amino acid that it changes to is very different in chemical properties, so that protein might not be able to carry out its normal function. A frameshift mutation is basically where a insertion occurs or a deletion occurs of a nucleotide or a sequence of nucleotides that might affect the reading frame of our sequence. So you can see here the addition of an A has resulted in all of the amino acids after that mutation being affected. Okay, and a block mutation is where instead of just looking at one nucleotide, there will be segments of whole chromosomes that might be impacted. Okay, so there could be inversions where your chromosome might rotate 180 degrees. There could be deletion of part of a chromosome. There could be a duplication, so a double up of a part of a chromosome. And there can be what we call translocation, so moving to a totally different chromosome as well. Moving on, so we also talk about selective breeding, which is a result of manipulating gene pools, really. So selective breeding is an example of what we call artificial selection because it involves human intervention. So things like the dog breeding industry would be a really good example of a selective breeding program. Oh my gosh, that animal's so cute, so we're going to add or breed this particular animal with this particular dog because we want these features to be shown. This is also prevalent in the dairy industry. So looking at the meat and dairy industry altogether, we could say like the production of meat that we're producing. We want low fat carcass. We want higher milk yield or better quality milk. Wool is another example as well. So it's where we know that there's variation in a population, but we might induce a selection pressure or we might have a favorable trait that we want to select. And so we breed those particular animals that might have that selected trait that we want. So we're increasing the number of animals that have that when manipulating the gene pool, and we're selecting the traits that we want. So survival and reproduction can really be at risk through these selective breeding programs. And they don't necessarily equip the animal for survival and reproduction in the world. It's very much what's going to help us. And so it can affect the survival rate of a population as well and cause a reduction in that gene pool. And finally, we talk about bacterial resistance and viral antigenic drift and shift. So this is basically looking at the consequences that might result because and ongoing challenges with treatment with pathogens and things like that as well. So looking here in terms of the development of an antibiotic resistance bacteria, there is variation in that bacteria population, and there might be certain bacteria that might be antibiotic susceptible, and there might be an ability for those bacteria to be resistant, okay, as well. Then there is some sort of selection pressure, so it might be the exposure to an antibiotic, and that might kill off all of our bacteria that are not resistant to those bacteria, or most of them. And then those ones that were resistant, they are able to continue to grow and develop and breed. Binary fission occurs where they replicate. And then now in that population, we have a lot of bacteria that are resistant to that antibiotic. Okay, and they're able to continue and replicate and pass on that resistance allele to other bacteria. And so that increases their allele frequency, which means that we have to find a different antibiotic and different treatments for that as well. If you have any questions regarding any of these dot points that are related to this aspect of the study design, please leave them in the comments below, and I'm happy to answer any last minute questions that you have as well. Thank you very much. Bye.